So now let's begin to look at each of the 4 sections for the complete oxidation of glucose. Again, we'll bring in with glycolysis, which is the splitting of glucose. We'll move into pyruvate oxidation. Then we look at the Krebs cycle. And finally, we look at the electron transport chain. Before we begin, let's orient ourselves, give us some perspective of where these things happen. Glycolysis occurs out in the cytoplasm of the cell, outside of the mitochondria. When we get into pyruvate oxidation, it happens in the mitochondrial matrix. The mitochondrial matrix is the very inside of the mitochondria. The Krebs cycle also occurs inside the mitochondrial matrix. And the electron transport chain will occur crossing the inner mitochondrial membrane. So across this membrane. For each of these different processes, there are 3 things that we're going to track. The first of which are the carbons. In addition to carbons, we're going to track electron carriers like NAD and FAD. And the third thing that we'll keep track of are ATPs. So always remember, these are the 3 things that we must track through each of the different phases of the oxidation of glucose. Let's begin by looking at glycolysis. Again, glycolysis. So it makes sense that we're going to split a 6-carbon sugar into 2 3-carbon sugars. This is a 10-step biochemical pathway. Let's take a quick look at the whole thing. These are each of the 10 different steps. Now you can look at these and become overwhelmed, but it's okay. You don't need to know each of those 10 steps until you take a course in biochemistry. For now, we're going to simplify the process. Remember that glycolysis occurs in the cytoplasm. We're going to see that there's a net production of 2 ATPs in this stage and 2 NADHs. Let's take a little bit of a closer look. In the first portion of this process, we'll see that we have to actually invest a couple of ATP to get things going. This is somewhat like adding a spark to start your fire. A little bit of energy has to go in order to get more energy out. So I call this first phase the energy investing stage. In the next stage, we're simply going to split this 6-carbon sugar into 2 3-carbon molecules. In the third phase of glycolysis is when all the fun stuff happens. This is where the 3-carbon molecules become converted into pyruvate. So we get 2 pyruvate out for each glucose. We'll also see that we get 4 ATP for each glucose. And we'll see 2 NADH for each glucose. So in this whole process, we began with a 6-carbon glucose. It's now been split into 2 3-carbon pyruvates. We have a total of 4 ATP produced and 2 NADHs. So we've tracked all 3 components that we need to track. Carbons, ATPs, and NADHs. So overall, in the glycolysis pathway, we have a net of 2 ATP because we invested 2, we got 4 out, and that equals a net of 2. The gross production was 4 ATP. So now, let's take a moment to diagram this, because after all, you're going to need to do that for your diagramming assignment this week. My diagram will be significantly more simple than that in the text. I have our uphill or energy investing reactions, followed by the downhill or energy revealing reactions. Next, I want to consider my carbons. Carbons in the beginning are in the form of glucose, and the glucose molecule has 6 carbons. At the end, these carbons have become pyruvate. We will have 2 times a pyruvate. And pyruvate is each 3 carbons, so we still have 6 carbons total. Next, let's consider our ATPs. We know in the energy investment stage, we had to put in 2 ATPs. and that gave us out 2 ADPs. And on the way down, we ended up having 4 ATPs produced. And in order to get 4 ATPs out, we had to put 4 ADPs in. The third component that we need to track are NADs. And in the downhill reactions, we'll find that there are 2 NADHs produced. In order to get those, we put in 2 NADs. So that's basically it. That's all we need in our diagram about glycolysis. We've tracked our carbons, we've tracked our ATP, and we've tracked our electron carriers. Remember, this NAD has come in to pick up 2 electrons and 2 hydrogens. Those electrons are now off in the carriers to go to the electron transport chain. And let's take a look at the more complicated version. You just need to pick out the components that we care about. That's where the ATP is used, where ATP is produced, where we find NADH, and where do all the carbons go. You can see that it's significantly more complicated than that. However, you'll be happy to know that you won't be held responsible for all of this detail. Let's take a look at an animation that should help us understand glycolysis a little more clearly.
The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a 4-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of 2 distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, 2 ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the 2 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins.
In order for glycolysis to continue and not run out of electron carriers, NADH needs to be recycled to NAD+. It can be recycled in one of 2 ways. Either it can be recycled in the process of aerobic respiration. When oxygen's available as the final electron acceptor, the electrons are dropped off by NADH, and then NAD is recycled. This produces a significant amount of ATP. However, if there's no oxygen present, then we're only left with fermentation or anaerobic respiration, where a molecule other than oxygen is the final electron acceptor and allows NAD to be recycled in order to continue the process of glycolysis. So, I have a little bit of an analogy here. It's kind of a far-fetching one, but imagine this. NAD are like limousines, and we're going to the prom. Electrons are like girls, and the protons or hydrogen ions are like the boys. And usually, couples go together to the prom in a limousine. So that would leave us with 2 electrons and 2 protons being carried by the NAD limousine. Now, when kids are going to the prom, they're really excited. So the electrons and the hydrogens are really excited. They're in a high energy state. They've been picked up at their house, which is glucose. And now they're going to be driven off to the prom. Now, there are many other kids going to the prom. So NAD limousines are going all over the place throughout the process of cellular respiration to pick up very excited boys and girls on their way to the prom. Once the limousines have dropped them off at the prom, then they can return to go and pick up other kids. But if we don't drop off any kids at the prom, then there's no way that we can have enough limousines in this town to pick up all the kids that need to make it to the prom. So in that sense, the NAD limousines need to be recycled. They need to drop off their H's and their electrons in order to become NAD plus and go back up into glycolysis and pick up more excited electrons and protons. The prom story will be continued. Back to real life. This leaves pyruvate with several different options. Either pyruvate will continue on down through those 4 processes of complete oxidation of glucose and cellular respiration in a process called aerobic respiration. Or if there's no oxygen, then NAD limousines need to drop off their hydrogens and electrons elsewhere. They'll do that in the actions of fermentation. If you're a person, that'll happen as the formation of lactate. So glycolysis is a very fast-acting system, and when we first start exercise, it's the system that's working to generate as much ATP as possible, although you've learned it's not very much, it's only 2 ATP. And what happens as a byproduct? When you first exercise, you get some soreness in your muscles. This is because of lactic acid. When lactic acid builds up, it makes the muscles feel fatigued. After all, it is an acid, and it's changing the environment so that the enzymes can't work as efficiently. So this lactic acid production can only go on for so long, but what it does allow is for NAD to be recycled so it can go back and pick up excited electrons and hydrogens from the process of glycolysis in order to keep glycolysis going and producing even just the little bit of ATP that it does. Now, if you're a yeast, then you would recycle these by producing ethanol instead of lactate. They have a different enzyme than us, so they'll turn pyruvate into acetaldehyde and then ethanol. This is the ethanol that we find in our beer and wine through the fermentation actions of yeast. Either way, the same thing. We're unloading NADH limousines to reveal empty limousines that can now go back over to glycolysis and pick up more excited electrons. I sort of make this as an analogy as the stop at the beer shop. But they're sort of low-budget limousines, so while the kids are stopped at the beer shop or the wine shop picking up some extracurricular products that you're not supposed to take to prom, then the limousines go off and pick up some more kids over in the glycolysis neighborhood. So with oxygen, pyruvate can go on into the rest of the reactions that we'll look at in this chapter that constitute cellular respiration. However, without oxygen, the only way to recycle NAD in order to keep glycolysis going is to produce lactate if you're a human, and if you're a yeast, to produce ethanol. This production of lactate and ethanol releases the limousines, or the NAD pluses, so they can go back into the process of glycolysis and continue to pick up electrons. Because if there's no one to pick up electrons, then no ATP can be made. So it's really important that NAD is recycled. Let's take a quick card quiz. Which of the following processes uses inorganic molecules as the terminal electron acceptor? Lactic acid fermentation is an anaerobic process. Is this true or false? Which of the following is classified as an autotroph? And during a heart attack, blood flow to the cardiac muscles is restricted. How would this affect cellular respiration? Remember, the blood is what carries the oxygen. Glycolysis costs this many ATPs, but makes blank ATPs, and thus it has a net yield of blank ATPs. All of the glycolysis reactions do not require oxygen and can take place in an anaerobic environment. All the glycolysis reactions. Is this true or is it false? Here are your answers.